structure of DNA. James Watson and Francis Crick elucidated this structure. James Watson and Francis Crick. Okay. And DNA chain consists of two strands of polynucleotides which are coiled each other in the form of double helix. Okay. So two strands of polynucleotides, okay, which are coiled each other in the form of a double helix. Okay. Next point is each strand are connected by phosphate ester bonds. Okay, phosphate ester bonds. Each strands are connected by phosphate ester bonds, and this is the backbone of each DNA from which bases are extended. Okay. Next, bases project each other in the structure. Okay, sugar and phosphate components. Sugar and phosphate components forms the structural frame. Structural framework. Okay. And bases are projected out. Okay. And the next point is these bases are held by hydrogen bonding between them. Okay. Bases are held by hydrogen bonding. Okay. Hydrogen bonding between them. Now the bases present in DNA are adenine, guanine, cytosine and thymine. Okay. And these bases are very specific. Only one mode of pairing with other takes place. Okay. Only they are very specific. Only one mode of pairing. Okay. Takes place. One mode of pairing with other bases is taken. For example, adenine. Adenine pairs only with thymine. It is specific. Okay. This is through via two hydrogen bonds. Okay. Next, guanine. Guanine with cytosine. Okay. This is specific via 3 hydrogen bonds. Okay. 3 hydrogen bonds. Other combinations are less favored. Okay. Now, this is the structure of DNA. Okay. It's a double helix structure. And guanine is pairing with cytosine. It is specific through 3 hydrogen bonds. Okay. And adenine with thymine through 2 hydrogen bonds. Okay. And the distance between here to here, that this is 34 angstrom, and between these two pairs is 3.4 angstrom. Okay, this pairing is specific, and all other pairing, all the other combinations are less favored. Okay, this is the double helix structure of DNA. Next, important functions of DNA. DNA is the constituent genetic material. It is the constituent genetic material of cell nucleus. Okay. Next, it controls the synthesis of RNA. It controls the synthesis of RNA, which synthesizes proteins. Okay, so RNA synthesizes proteins. Okay, so the message for the synthesis of particular protein is present in DNA. Next point, it is a unique property of self-replication. Self-replication. Self-replication means during cell division, an identical DNA strand is transferred to daughter cells. Okay, DNA strands are transferred to daughter cells. Okay. So, during cell division, an identical DNA strand are transferred to daughter cells. Okay, so this is responsible for the maintenance of heredity from one generation to another. Okay, so this DNA are responsible for the heredity from one generation to another generation. Okay, and also we know that DNA consists of different bases like adenine, guanine, cytosine and thymine. They are held together through different hydrogen bonds. Okay. RNA. RNA is ribonucleic acid. Okay, and the sugar present in it is beta D ribose, and it consists of the bases adenine, guanine, cytosine and uracil. Okay, and they are denoted by A, G, C and U. Okay, it is in contact with DNA and the sequence of bases depend upon the sequence of bases in DNA. Sequence. Here the sequence of bases in RNA depends upon the bases, the sequence of bases in DNA. Okay. Next point. Next, it is of three types which play an important role in the protein synthesis. These are first one, messenger RNA or mRNA. Okay. So now, what is messenger RNA? It carries the messages of DNA for specific protein synthesis. It carries the message of DNA for specific protein synthesis. Okay. Second one is ribosomal RNA. Ribosomal RNA. Ribosomal RNA means it provides site for protein synthesis in the cytoplasm. It provides site for protein synthesis in cytoplasm. Okay. Next, third type is transfer RNA or tRNA. Okay. Now, it transfers amino acids to the site of protein synthesis. This transfer amino acids to the site of protein synthesis. Okay, these are the three types of RNA. Next, there are about 20 different types of tRNA. So, 20 different tRNA molecules in cytoplasm. Okay, 20 different tRNA in cytoplasm, and each is specific for one amino acid. Okay, so each is specific for one amino acid, and RNA guides the biosynthesis of proteins. It controls the biosynthesis, or it guides the biosynthesis of proteins. Okay, and controls the process of learning and memory storage. Okay, controls the process of learning and memory storage. Okay, and this RNA contains only a single strand. It is a single stranded structure. Okay.